Okay, so this is Lesson 9 Group Problems. I'm Sto Rosello.
I'm Valerie Wendell.
And we're just going to go 1 through 5, just alternate back and forth. So we'll start with 1 and then move on.
Okay, so hopefully you can see this. Oh, I forgot to put our origin. Or, see, this is our origin of replication. Oink. There, okay. So that is that. And so we have our two, this is kind of messy. So we have our two leading strands here, and then our lagging coming up behind it because the leading strands start and they are continuous, and then the lagging start behind the leading, and then they have to, as like the opening gets bigger, the lagging backs up. And so there's, we have our RNA primase, which are these red, are they RNA? Yeah, primers, and these are the red boxes, and so that's where the replication starts. And so we have the leading, and then for the lagging, with every group, it starts over and over again, and that's where you get your Okazaki fragments on the lagging strands there. And then you have your DNA helicase here, which opens up the DNA sequence. And then you have the DNA primase here, and that is what shows the RNA primers where to start, where to go, and so we have those there. And then we have our DNA primase 3 here, which links the nucleotides into one strand, and then the DNA polymerase 1, which does the same thing, but it also kicks off the RNA primers. Did I get everything?
I believe so.
Okay.
Okay. So, it says also to indicate the polarity of all nucleic acid strands.
Yeah, so, okay, so for the parent strand, we have this, this bottom strand going from 3 to 5, but then it is being synthesized from 5 to 3, and then here we have this going from 3 to 5, and then it's being synthesized from 5 to 3. So those are the polarities, sorry about that.
Okay. So for number 2, we have that one right here. It says, in the blanks provided, indicate the protein that best fits the description. For some questions, there can be more than one correct answer. So these are the ones that we got. If you got something that's similar to that, then I guess that works too. But for A, the protein complex that synthesizes primary in eukaryotes is DNA primase. Sometimes we write just primase or ligase or gyrase, and in all of the cases here that you'll see it's DNA before that, so DNA primase. Okay, for 2B, it says this protein promotes the separation of the AT-rich sequences within the ORIC, and we got DNA A for that one. And for C, this subunit of DNase polymerase 3 contains proofreading activity. So proofreading activity, the ability to go back and correct any issues that might have happened. This would be an epsilon subunit. For D, this protein synthesizes the leading strand in eukaryotes. For this one, we got DNA polymerase epsilon. There's a couple variations, 1, 3, epsilon, delta, alpha, so this one falls under the epsilon. For E, we got this protein displaces DNA polymerase 3 from the chromosomes to terminate replication, and we got that as the TUS protein. For F, we got it says this protein has a 5' to 3' polymerase and a 5' to 3' exonuclease activity. And exonuclease just means it can start on the ends instead of in the center. So for that one, we got DNA polymerase 1. For G, this protein introduces negative supercoils into the DNA ahead of the replication fork, and this would be your DNA gyrase. H, this protein binds directly to the ARS elements, which would be the ORC, not the Outdoor Recreation Center. For I, this protein prevents DNA strands from annealing during replication. And annealing is just basically having them come back together. So we don't want that to happen. So like if this is separated, we don't want that to close back up. and so that would be the SSBPs. For J, this enzyme seals Okazaki fragments into a continuous strand of DNA, and that would be the DNA ligase. So that's what we got for 2.
Okay. Alright, so for number 3, it says, so this is all about like the enzymes in the DNA. And so for A, it says list 5 enzymes, prokaryote or eukaryote, that consume ATP during DNA replication. And we got DNA helicase, gyrase, primase, ligase, and DNA A. Those all consume ATP during replication. And then for B, it says list 5 enzymes, prokaryote or eukaryotic, that consume DATP during DNA replication. And that is polymerase 1, 3, and then the subunits alpha, sigma, epsilon. Delta. Sorry, that's delta. I don't know my Greek alphabet very well. C, list 5 enzymes, prokaryotic or eukaryotic, that have 3 to 5 exonucleic activity. And so we've got polymerase 3 and 1, and then delta, alpha, and epsilon subunits. Again, same as B. And then D, indicate 1 enzyme, prokaryotic or eukaryotic, involved in replication that uses DNA as a template to synthesize RNA. And we got the DNA primase. And then E, indicate 1 enzyme, prokaryotic or eukaryotic, that uses RNA as a template to synthesize DNA, and that is the telomerase.
Okay, so for 4, so suppose you're trying to synthesize DNA in the lab. A single-stranded DNA molecule, which is given, will serve as a template for the reaction. This template is mixed with the DNA polymerase 3, hollow enzyme, a primer with the sequence, with this sequence right here. So the TGC, so on and so forth, and a buffer. So you decide to perform 4 DNA synthesis reactions. Determine the sequence of your DNA strand synthesized if you use the nucleotides described in parts A through D. So for A, B, C, and D, it will give you what nucleotides are available to be able to put into the sequence once your primer has attached to the parent strand that you're trying to synthesize. So assume that if the DNA polymerase cannot incorporate one of the nucleotides that's listed, the reaction is going to terminate. Do not include the sequence of the primer in your answer. So for 4A, the DATP, DCTP, DGTP, and DTTP are all the regular nucleotides that you would normally be placing into a replicated strand. So you should not have any issues in the replication, and so for that, once the primer is going to attach over here, the sequence will start once the primer, after the last nucleotide of the primer. So we have them listed 5' to 3', and for this one it should follow the entire sequence. So the parent sequence, it gives you it also in the 5' to 3'. We know that it attaches the opposite way, right? So the 5' will attach to the 3' end of the parent, and will end on the 3' with the 5' of the parent. So in this one, it's written we have the ACT, GAT, GG, TCC, TGC, TAT. So the whole thing should go through. The nucleotides in B, if you look, we have DCMP monophosphate, which is not usable in replication, and so the first time that you get to a C in your replication sequence, it's going to drop, which in this case, for B, we look at the original one, the second nucleotide is a C, and so that actually will never occur, and so your sequence should just be 1 nucleotide long. It's just going to be 5' A, 3'. For C, you notice that the UTP will be included. The other ones are all regular, so UTP is found in RNA, and so it won't be usable in this DNA sequence that we're trying to replicate. And U is replaces T, from DNA to RNA. So the first time that we run into a T, the sequence should drop. So looking at the parent strand again, we have A, C, T. The T will never happen. So for C, our answers should be 5', A, C, 3'. And then for D, the GTP is not a daughter nucleotide, and so that is something that will also never occur in the replication process. So the first time that you get to a G, it's going to drop off. So in this case, we have A, C, T, G. So as soon as we get to the G, we can go ahead and cut it there. So the answer should be 5', A, C, T, 3'. So that's how we rounded our answers for number 4.
Perfect. Okay, and then for number 5, we have a list of all the proteins involved in DNA replication. Then for A, it says, what of the above proteins is found in prokaryotes? And so we got TUS, polymerase 1, 3, epsilon, alpha, helicase, primase, DNA A, and gyrase. And that's the 5 we got for A. B, which of the proteins is found in the mouse musculus? And we know that the mouse is a eukaryotic, or has eukaryotic characteristics in cells, and so it will be everything except for the DNA A's. So all of that. And then for C, it says which of the above proteins is involved in chromosome compaction, and we got none, because everything is, all the protein involved in replication is meant to unravel the DNA and elongate it, no compaction. So that's what we got. Then for D, which of the above proteins is involved in the initiation stage of DNA replication? We got helicase, CDC6, and that's for the eukaryotic, and then DNA A for prokaryotic. And then for E, which of the above proteins is involved in the termination stage of DNA replication? So you have telomerase for eukaryotic, and then TUS for prokaryotic.
Those are the answers we got.
That's what we got. Thanks for watching.